I've been asked to sort of give an overview of basic immunology, and so I've tried to do it around the different types of vaccines that are administered to children and some of the elderly. So first of all, I just thought I'd give a little overview of how the immune system actually evolved. So as most people know, your body is covered in commensal or natural bacteria in the gut, on the skin, in the sort of nasopharynx, and this is one of the things that drives the development of a healthy immune system and sort of helps you to regulate responses to other pathogens. So why aren't we constantly ill if we're covered in all these bacteria all the time? So first of all, the body's able to defend itself against these commensal organisms because we're basically covered in this impenetrable layer of skin, and this prevents bacteria from getting into the system, so getting into the blood and the tissues. And things like mucus and saliva and tears and sort of sebaceous secretions on the skin help to protect it and keep it moist so that you don't get any breakdown in the integrity of it. And so if you get a breaching of the skin, so if you get a wounding or some kind of abrasion to the skin, this can trigger an inflammatory response. And inflammation is one of the main drivers of your immune response. So without any kind of inflammation, you won't get much of an immune response to anything. But this inflammatory response is what triggers the body to recognise that something bad's happening to your body. And one of the main roles of this system is a population of cells called phagocytes. And basically these cells, I don't think they actually look like that little alien thing on the side, but basically they act like the vacuum cleaners of the body, and they go around cleaning up invading pathogens and debris from infected cells. So hopefully they limit the amount of inflammation that goes on and clear pathogens before they can really take hold. And on the other side of that, you've got another part of the immune system that can generate antibody responses and T-cell responses, and that side of the immune system is the part that we try to target with vaccines, which I'll talk about in a moment in more detail. But this is the side of the immune system that generates what we call immune memory, so I'll explain more about that in a moment. So what do we know about age and immunity? For disease, Andy's probably going to hate these graphs, but anyway, for disease, there's sort of two peaks of infectious disease, and one of them is in the neonatal period, sort of within the first 2 months, and then this sort of incidence wanes throughout life, and then as you become older and your immune system starts to become a little bit tired, then you start to see a rise in infectious diseases again. The exception to that is in sort of teenage years, you also see this peak, or we used to see a peak in disease caused by meningococcus. So you'd get outbreaks of meningococcal disease. And one of the reasons for these peaks in disease are related to your development of immunity. And so in very young children, in the first couple of months of life, they're protected by maternal antibody that crosses the placenta. And that sort of helps protect the child during that first couple of months. But this wanes quite rapidly. And so you end up with this peak appearing in childhood where your antibody's waning and sort of disease incidence starts to rise. So, and then as you grow older, you develop antibody in response to natural infections. And so gradually this protects you and you see the drop in disease rates. But then as you get older, your immune system becomes less effective and less able to mount responses to new pathogens. And so then you start seeing this increase or decrease in antibody and a rise in disease. There's a whole lot of other things involved in that, but that's kind of the basic thing. And so we introduced vaccine at various different ages to try and sort of fill these gaps in the immune response. And some of these are quite effective and make antibody that lasts for a long time, and some just fill a certain time period and just help. So for neonates, get them through that first 12 months to keep antibody levels high enough to protect them while their natural immune response develops. So the immune system itself is divided into sort of two arms. You've got an innate immune system, which basically is developed by the time you're born, and then you have an adaptive arm, which develops as you get older, so in response to natural infections. So the innate immune system basically involves what I was talking about earlier, so the skin, the secretions at mucosal membranes like mucus, the phagocytic cells that gobble up bacteria, inflammation which helps trigger responses, and then a lot of other components which are involved in the healing process. And so you need an intact innate immune system to be able to mount other further responses. And then the adaptive arm of the immune system is divided into two, really. So you've got the humoral side, which is mediated by antibody, and antibody is produced by B cells, and hopefully you can make memory B cells that hang around, so next time a pathogen comes along, it remembers it and you'll be protected. And then you have a cellular side, which is more mediated by T cells, and these can be either helper T cells, which sort of provide help to other aspects of immunity, or cytotoxic cells, which are able to lyse infected human cells, so hopefully limit viral infections. And together, all of these help you make an effective immune response to a pathogen. But if any one of those components is missing, then that's when you get into trouble, really. And so, as I've said, these are some of the cells that are involved in the immune response. They all form part of what we call the white blood cells, and they're all derived from bone marrow. And so you've got B cells, which make antibody, T cells, which provide help to B cell responses, and also lyse target cells. You have NK cells, which are killer cells, and they form part of the innate immune response, so they can lyse infected cells even if you've never seen a vaccine antigen before or a pathogen before. And then on the other side of things, you've got the phagocytes, which sort of clean up after an infection. So you have a dendritic cell, which has these sort of funny arms all over it, and that just means it's got lots of surface area and also is able to get between sort of epithelial cells. You've got macrophages and neutrophils, and those ones, their main task is to just clear up debris and bacteria. So all of these immune cells are constantly recirculating. A lot of them reside in tissues if you're not having an immune response, but the B cells and the T cells particularly recirculate through the blood and the lymph. So they're constantly sort of surveying your body for infectious organisms. And we call this sort of immune surveillance. And so hopefully, wherever in the body you get an infection, these cells will arrive and try and clean it up. And then the phagocytes, they meet a bacteria or a virus, and then it binds to the cell surface, and they slowly engulf it. And then they have things inside the cell which can break it down, and so it doesn't escape back into the body, and it's just completely cleared from the body. And so the next thing to talk about is sort of where the sort of main immune responses take place. So we've talked about the blood and the tissues are sort of the areas where things get picked up, and the sort of paths where things sort of travel to get to the lymphoid system. And it's the lymph nodes and the spleen and the mucosa which are really sort of the driving force, or the sort of home of the immune responses. And so the body's sort of covered in lymphoid tissue, really. But the ones we try and target most are sort of respiratory infections, which the immune responses tend to occur in the adenoids and the tonsils. Blood infections, the spleen's really important for blood-borne infections, and it cleans the blood, and there's cells in the spleen which help to mount immune responses. And gut infections, so most of your gut has got lymphoid tissue all the way along it, and so any bacteria that invade through the gut get picked up by the lymphoid tissue in the gut. And then with vaccines, mostly what you're doing, because they're administered through the skin, is you're targeting the draining lymph nodes from that side. So in the sort of armpit or in the sort of groin area, if you've had an injection in the leg, those are the sort of sites that we think the vaccine responses occur in. And so what happens is infectious organisms invade through the skin or the gut. The immune cells pick these bacteria up, take them back to the lymph nodes, you get a response, and then this then sort of re-spreads around the body and fights infection at local places. And so I'm just going to go a little bit more through what happens with a natural infection now, so it kind of makes a bit more sense. So with a natural infection, I'm using an example of a viral infection at the respiratory airway, just as one example, but a similar thing would happen at the gut mucosa as well. So the airway mucosa itself is made up of epithelial cells, which have cilia on the surface, which are like little protrusions which are constantly moving, and the mucus as well, which help basically prevent pathogens from attaching, and it's constantly moving, so it's constantly ejecting pathogens. But occasionally you'll get a virus which can bind to a receptor on a healthy cell, and this then allows it to invade the healthy cell. For viruses in particular, they need to get inside a human cell so that they can replicate. And so as soon as they're inside the cell, they start to multiply. And this triggers actual destruction of the host cell eventually, because it overrides all the cell mechanisms. And this triggers various responses, but one of the main things is inflammation. And the first thing with inflammation is you start to get sort of leaky blood vessels, and the cell integrity starts to break down, and so you get components of the blood starting to leak into the tissues, and that's what causes like the redness and the heat and the sort of swelling at an infection site. But the other thing that happens, and the little blue arrows, I don't know if you can see those properly, the infected cells start sending out signals to the immune system, and these can be chemicals that go into the blood and attract immune cells to the infection site from anywhere in the body. The cells are able to sort of follow these chemicals through the blood and find the infection site. So what happens is you get sort of phagocytes and T cells and B cells and the dendritic cells with those funny arms all coming to the infection site. And so you end up with this sort of infection site that's full of sort of blood serum proteins, and the cells are able to sort of squeeze through the leaky blood vessels into the tissues, and then they start to fight the infection. And so the first thing that happens is you get antibody made. I don't know how clear this shows up actually. There's little green antibody molecules there. And the other thing that happens is some of these phagocytes actually pick up some of the viruses. So sometimes they're engulfed and destroyed, but the dendritic cells with the funny arms, they actually pick up the bacteria or the virus, and then they can transport that back to the lymph node, which I'll show you in a second. But the main purpose of the antibody that you make in this initial response is to coat pathogen that's either in the blood already, or it can get across the epithelial cells to the mucosal surface and coat new virus that's at the cell surface. And that can prevent attachment to epithelial cells so it doesn't invade, but it also targets it so the phagocytes pick it up and eat it. And that is basically what forms your innate immune response to an infection. And if it's only a low-level infection, it will clear that up and you won't even really worry about it. But at the same time, the dendritic cell takes the virus that it's picked up back to the lymph node, and then at that point is where your adaptive immune system takes over. So these cells that are stimulated sort of proliferate and multiply, and then they migrate back to the blood when you re-encounter the infection, and you'll get more antibody produced. But what you also get is T cells that can recognise infected cells and then will lyse it, which means the virus is then exposed to phagocytes that can gobble it up, and also you've limited the amount of infection that will happen in your mucosa. And so hopefully that sort of gives you a sort of summary of what happens with a natural infection. I'm actually taking more time than I thought with this. So how do we mimic this with a vaccine? So for your ideal vaccine, what you want is a rapid antibody response to mop up any invading pathogens, but you also want to induce memory B cells and T cells that will fight an infection next time you meet it. And so this was what we talked about earlier with the immune system. So mostly what we do is we target the adaptive side of the immune response. So our vaccines contain either whole bugs or pieces of the bacteria or virus, or sometimes toxoids like tetanus or diphtheria. And we just inject those and hope for the best that something will happen with your antibody and your T cells. And the other thing we do to try and enhance all of this is, you get your antibody and your T cells. The other thing we do to enhance all of this is to use adjuvants. So most of the vaccines have alum-based adjuvants, but some of them are now starting to get new sort of level adjuvants, and one of them is MF59, which has been used in a flu vaccine. And these target the innate side of the response, and so they can trigger things like inflammation and phagocytes to get involved. And so together, those two aspects then enhance your immune response to the vaccine. But as you know, we have several different kinds of vaccines. You have live vaccines, you have vaccines that are made up from components, and then we have conjugate vaccines as well. And so I thought I'd just hopefully quickly give you a quick overview of what happens with the different sorts. So if you remember what happened with a natural infection, a live attenuated vaccine kind of almost replicates this, except that a live attenuated vaccine during its manufacturing process has been it's been manufactured so it can't replicate as effectively in host cells or can't spread and invade new host cells. And so what happens when you give an attenuated vaccine, so I've given some examples at the top of these sort of vaccines, what happens is you'll introduce virus into the skin, so the needle's sort of breaking that first innate barrier anyway, and then you're introducing the virus into the skin, which can still replicate and cause a very low grade infection. And so what happens is you get a similar process as to natural infection. So you'll get some inflammation and you'll get some leaky blood vessels, and then you get recruitment of your immune system as we saw with the natural infection. So you get your cells and your antibody arriving to the injection site. And then again, your cells start to make antibody, your phagocytes gobble up some of the pathogens, and again, some of this gets taken back to the lymph node. So you're again getting sort of an innate response to your vaccine, and also hopefully making a memory response so that you're protected against whichever virus you've immunised against. And so hopefully next time you'll get a more rapid response. Yeah, that's what I just said really. But I've skipped through it a little bit. The other sort of vaccine we have is a killed vaccine or component vaccines. And what I mean by components is where we take like proteins from the cell surface or the capsule. The examples I've given at the top are all protein-based vaccines or killed viruses. Someone will correct me if I'm wrong. And again, these are introduced into the skin, but the difference between this and a live attenuated or a natural infection is that these viruses can't replicate, or obviously the proteins can't replicate. So they're actually quite inert on their own and don't induce a very good response. And so the way around that is to add these adjuvants that I mentioned earlier. And so basically what happens is your particles or your components are absorbed onto an adjuvant, which makes it bigger and more attractive to the immune system. And this works in two ways. One thing is that adjuvants are actually able to stimulate the phagocytic cells to come to the injection site. So this means that you'll get your antigen picked up by the dendritic cells, and you'll get sort of initial antibody response to the particles, which you would have otherwise not have got if it was just without the adjuvant. And then you get the same process again where you get antibody made, and then you get your innate response that protects reinfection at the mucosal surface. But then you still get this memory response in the lymph nodes. And so adjuvants are really good at enhancing memory responses to virus, proteins and killed viruses and bacteria. Sorry. And then the final one is the conjugate vaccine. So these include vaccines like pneumococcal, conjugate vaccines, so Prevnar, Hib vaccine, Menc, and the Men-ACWY, which I haven't actually included up there. These vaccines are all basically made up from the capsule of a bacteria, so the coat that surrounds it, which is completely, really inert to the your immune system doesn't really recognise it. And in children under 2, they can't respond to these kinds of antigens at all. And so what they've done to get around this is conjugate the polysaccharide, which I've made orange in this case, to a protein that we see all the time, so something like tetanus toxoid or diphtheria toxoid. And so you get this, what we call a conjugate, and the protein and the polysaccharide are linked together. And because you're immunised against things like tetanus and diphtheria all the time, you've already got some of this immune memory to help you respond then to this sort of combined antigen. So what happens with this vaccine is basically your vaccine antigen gets picked up and taken back to the lymph nodes. And then it does two things. So first of all, your B cell will bind the polysaccharide component And the B cell will then be specific for your bacterial capsule. So its antibody that it's going to make is specific for that capsule. But at the same time, it takes up that whole conjugate complex within itself. And then it's able, I don't know how clear this is actually, it's quite small. It's able to actually then load the protein component, so the conjugate component, onto its cell surface. And a T cell is then able to recognise that and bind to it. So the two cells basically bind together and they sort of send messages to each other which enhance the response to the polysaccharide component of the vaccine. And we call this T cell help. So this is all going on in the lymph node. So at this point, again, your B cells will then proliferate and they're all specific to your polysaccharide component. And so any antibody that gets produced then is specific to the polysaccharide. So what happens then when you get reinfection is you've then got antibody specific to the capsule. So if the bacteria manages to invade, it's targeted, it coats the bacteria and targets it for phagocytes, but it could also help to block it actually entering the body at all by blocking it at the mucosal surface as well. And so all of that in the end, what are we aiming for with all these different immune responses? So we have, most of our sort of vaccine schedules consist of a prime and then a boost. And so this really is an example of an infant vaccine. So in the first few months of life, you've got your maternal antibody, and what we're trying to do is enhance that. So you give a vaccine, and it could be 1, 2 or 3 doses during that time, but you get your first antibody response, which gives you this nice peak of antibody. But depending on the vaccine, it may wane really quickly, or it may last for quite some time. And so the other thing is that you get a lag phase in the response. So because it's a first response, the immune system takes a little bit of time, so before you start to see this rise in antibody. So then you come back and you give a booster vaccine, and some of them are component vaccines, so there might be more than one antigen. So the response to your vaccine A component will be a booster response, so it'll be more quick and it'll be higher, so protects you instantly, well not instantly, but much more quickly than the primary response. But your response to the second component will look like the primary response, so you've kind of got two things going on at the same time. But the lag phase to the primary response is still a week-ish, whereas to the booster vaccine, this is reduced down to a few days, and so you've already enhanced the immune system because you've got this more rapid response to your booster vaccine compared to your priming. So really what the priming does is help get you through the first 12 months while your immune system is maturing. And so in summary, what we hope to do with vaccines is mimic natural infections and make immune responses that are rapid and long-lasting and immunogenic in all ages, so from neonates to the elderly. So I hope that was clear, and that's all.